We are taking a saline solution and rinsing our mouth to contain cheek cells, cells which are the smallest unit of an organism. Now we are placing 1 milliliter of the cheek solution in a tube, which we will then place in the centrifuge. We just finished the spinning of our cells in the centrifuge. This white clump at the bottom of the tube is a pellet of concentrated cheek cells.
The cheek cells are then transferred to a microcentrifuge tube containing Instagene matrix. It is important to put them in the Instagene because it is made up of negatively charged beads that grab certain ions out of the solution, which stops the enzymes from degrading the DNA. Go.
After cells have been transferred into the Instagene matrix, the suspended cells will need to be incubated at 56 degrees Celsius for 10 minutes. Halfway through this incubation process, the tubes will be shaken and then placed back into incubation for the remaining 5 minutes. This allows the plasma membranes to soften and clumps of cells to separate from each other. The hot temperature deactivates enzymes that can degrade DNA, which is an important step in the preservation of DNA templates for further investigations.
Okay, we just finished incubating our tubes at 56 degrees Celsius. Now we are going to incubate our tubes in 100 degrees Celsius. In our tube, we still have the Instagene matrix and our cells that have been softened. In this next process, our cells are ruptured by the heat and all the cellular contents like DNA and enzymes are released. The enzymes released will typically chop up the DNA, but since we still have the Instagene in our tube, it will keep the DNA safe. The extracted DNA will then be used as a template for our PCR amplifications.
Go.
Now we place our tubes in the centrifuge, which spins them for 5 minutes. This separates the DNA liquid from the Instagene based on density. Separating allows us to prevent getting the Instagene matrix in the PCR reaction, as that would inhibit the reaction.
Shaking.
Okay, so once we have our DNA into our PCR tube, we add the master mix. The master mix contains 3 things: nucleotides, which are the bases of DNA. They make up the middle parts and have different names like A, T, C, or G. It also contains primers. Primers are the complementary nucleotide sequences that bind to the piece of DNA where we want to start copying. It also contains DNA polymerase, which is an enzyme that reads the DNA sequence and adds the complementary nucleotides to build a new strand.
After the addition of the master mix to the PCR tube, it is then placed into the thermal cycler. This is when the PCR reaction will take place. While in the cycler, the temperature is raised to 94 degrees Celsius for 2 minutes, and then
Another minute.
for 3 minutes?
Mm-hmm.
Okay. And then to 60 degrees Celsius for 1 minute, and then back up to 72 degrees Celsius for 2 minutes. These steps are repeated 40 times, and then the temperature is back down to 72 degrees Celsius for 10 minutes, and then 12 degrees Celsius until needed, right?
Go.
Polymerase chain reaction, or PCR, is a technique used to make multiple copies of a segment DNA of interest, generating a large amount of copies from a small initial sample.
So the first step at 94 degrees Celsius, maturation happens, the process of separating the double-stranded DNA template into 2 single strands. The second step at 60 degrees Celsius, annealing occurs. The primer anneals or finds the complementary sequence on the second single strand template strands of DNA. They are all called primers because they prime the synthesis of a new strand. The third, at 72 degrees Celsius, extension happens. This is where the Taq DNA polymerase adds nucleotides A, T, G, and C one at a time to the primer to create the complementary copy of the DNA template.
This reaction is repeated 40 times, thus generating billions of copies of DNA of a particular region of a genome.
So we've loaded our samples into the thermal cycler, and now it's running the PCR reaction.
PCR can help detect mutations that can cause genetic disease and viral infections. It allows a gene to be researched, permits gene therapy, and proteins can be produced from the gene. PCR is used in forensic science. It helps criminal investigators to identify suspects, allows genetic fingerprinting, etc. PCR was used to find missing victims that were in the World Trade Center on 9/11.
It's a really useful process, a way to make billions of copies of a segment of a genome in a really short period of time.